if I told you that? With the nucleus of every cell lies a blueprint so intricate. It holds the key to the very essence of life itself. As we embark on our journey through the central dogma of molecular biology, let's pause to reflect on the minds that paved the way for our understanding.
Some scientists once believed that the central dogma of biology was set in a stone. DNA makes RNA. RNA makes protein in a one-way street. However, the groundbreaking work of Rosalind Franklin, along with the collaborative efforts of Francis Crick and James Watson, shattered this illusion. This process is described in the central dogma of molecular biology. It's like the rulebook that guides our cells in making proteins, the building blocks of life.
Let's begin by revisiting the star of the show, DNA. Found in the nucleus of every cell, DNA contains the instruction for building proteins that carry out various functions in the body.
The central dogma consists of three key processes: replication, transcription, and translation. Replication is the first step where DNA makes copy to ensure that each new cell receives a complete and accurate set of genetic information during cell division. This process maintains genetic stability and allows for the transmission of genetic traits from one generation to the next generation.
This remarkable process occurs in the nucleus, where enzymes unwind the DNA double helix and synthesize new strands of DNA, using the existing strands as templates. The end results of two identical copies of the original DNA molecule.
With DNA replication complete, we move on to transcription, the second step of the central dogma. Transcription is the process by which the genetic information encoded in DNA is transcribed into a complementary RNA molecule.
This process also occurs in the nucleus, where transcription factors bind to enhancer and promoter regions on specific DNA segments, attracting RNA polymerase to initiate transcription. As the transcription initiation complex forms, RNA polymerase synthesizes a strand of mRNA or messenger RNA. This mRNA carries the genetic message from the DNA to the ribosomes in the cytoplasm.
Finally, we arrive at translation, the third and final step of the central dogma. Translation is the process of decoding the messenger RNA message into a sequence of amino acids, which are the building blocks of proteins.
In this step, the messages travel from inside of the nucleus onto the ribosomes in the cytoplasm, where they are ready to make specific proteins. The ribosomes act as the protein-making factories, where they decode the mRNA sequence and link together the amino acid brought by the transfer RNA or tRNA molecules.
As the ribosome moves along the mRNA, it reads the mRNA in triplet codons, starting with the AUG start codon. Codons are sequence of twin nucleotides encoding specific amino acids.
Exactly, and let's not forget the importance of nitrogenous bases in this process. Nitrogenous bases such as adenine, guanine, cytosine, and uracil are the building blocks of RNA. They form base pairs, adenine pairs with uracil in RNA, and adenine pairs with thymine in DNA.
This base pair determine the sequence of codons in mRNA, which in turn dictate the sequence of amino acids in a protein. The ribosomes then link the amino acid together to form a polypeptide chain, which eventually falls into a functional protein.
To kick things off, let's explore the enzymes that catalyze these processes. Let's dive into DNA ligase, DNA polymerase, RNA polymerase, reverse transcriptase, and ribosomes.
Each of these enzymes plays a crucial role in maintaining the integrity and flow of genetic information. First, we have DNA ligase, often referred to as the molecular glue. DNA ligase plays a crucial role in sealing the gaps in DNA strands during replication and repair.
Secondly, DNA polymerase ensures accurate DNA replication by synthesizing new DNA strands complementary to the template strands. while RNA polymerase transcribes DNA into RNA. It reads the DNA template and synthesizes a complementary RNA strand known as messenger RNA or mRNA.
And don't forget reverse transcriptase, which is a unique to retroviruses and help in synthesizing DNA from RNA. It's a fascinating enzyme that plays a critical role in the life cycle of retroviruses. Now, let's introduce our viral element. Viruses such as retroviruses hijack host cell to replicate. They utilize RNA as their genetic material.
In the case of viruses, the mRNA produced during transcription serves as the template for protein synthesis. This brings us to our final destination, translation.
Now that we have discussed the sequential flow in processes involving central dogma and enzymes that catalyze these processes, let's move on to the essential components required for the central dogma to occur. Nucleotides, amino acids, and ATP are the key players here.
First and foremost, we have nucleotides. These are the building blocks of DNA and RNA, containing the genetic information necessary for coding proteins. During transcription, RNA polymerase utilizes these nucleotides to synthesize the complementary mRNA strand.
Next, let's explore another essential component in the central dogma, ribosomes, composed of ribosomal RNA and proteins. Ribosomes are cellular component critical for protein synthesis during translation. They read the mRNA sequence and catalyze the assembly of amino acids into polypeptide chain, ultimately forming functional proteins.
Amino acids are the building blocks of proteins. During translation, ribosomes read the mRNA sequence and assemble amino acids in the correct order to form a protein. The sequence of the amino acid determines the structure and function of the resulting protein.
Last but not least, we have ATP or adenosine triphosphate. ATP serves as the energy currency of the cell, providing the energy needed for various cellular processes, including those involved in the central dogma. Without ATP, these processes would not be able to proceed efficiently.
And so, as we conclude, let us carry forward this understanding of the central dogma.
Not merely as a series of molecular processes, but as the cornerstone of our comprehension of life itself.
We hope that this session had shed light on the fascinating world of molecular biology and the central dogma.